At the foundation of multicellular life lies the eukaryotic cell. Inside the nucleus of these cells is the genetic code for an organism organised into chromosomes. The life of a cell is governed by the cell cycle. It consists of interphase, encompassing the G1 phase mainly for cell growth, S phase for DNA synthesis, and the G2 phase, where the DNA is checked for any mistakes, followed by mitosis and cytokinesis, which are vital for the division of somatic cells. Mitosis commences with prophase, where the chromatin condenses down into visible chromosomes. The nuclear membrane breaks down and centrosomes migrate to the opposite poles, forming the mitotic spindle, composed of microtubules. During metaphase, spindle fibers emanating from the centrioles attach to the kinetic cores of each chromatid, and chromosomes align at the metaphase plate, the central line of the cell. In anaphase, spindle fibers shorten, pulling the sister chromatids apart toward opposite poles, ensuring each new cell will receive an identical set of chromosomes. In telophase, chromatids arrive at opposite poles. Nuclear membranes reform around each set, creating two distinct nuclei, and the chromatids begin to decondense back into chromatin. Following mitosis, the cell divides during cytokinesis. In animal cells, a contractile ring composed of actin filaments constricts the cell membrane at the equator, pinching the cell into two separate cells. The result of mitosis and cytokinesis is two genetically identical diploid daughter cells. Each cell contains the same number of chromosomes as the original somatic cell. For prokaryotic cells, cell division is achieved through asexual reproduction in a process called binary fission. Despite precision, errors like non-disjunction can occur during anaphase, where chromosomes fail to correctly segregate, leading to cells with abnormal chromosome numbers known as aneuploid cells. Aneuploidy disrupts the balance of genetic material, leading to altered gene expression, as genes are either overexpressed or underexpressed. To prevent the replication of aneuploid cells, the body activates apoptosis, which is controlled cell death. However, aneuploid cells can sometimes evade apoptosis, leading to uncontrolled cell division and tumour formation. The development of cancer poses a severe threat to the organism's survival, as malignant cells disrupt normal tissue function and can spread throughout the body. Cancer often arises from aneuploid cells that have bypassed apoptosis. The imbalance in chromosomes can activate oncogenes or deactivate tumour suppressor genes, leading to unregulated cell division. Because of this, tissue made up of aneuploid cells can grow much faster than normal tissue. Tumours formed by such cells can invade tissues, disrupt organ function, and evade the immune system, posing a significant threat to the organism's survival. Aneuploidy in the germline affects the formation of cells used in sexual reproduction called gametes. When incorrect segregation occurs in mitosis to create the germline cells, the resulting daughter cells will have 47 and 45 chromosomes. If these aneuploid germline cells then go through meiosis to produce gametes, they will create gametes with either trisomy, where the cell has an extra chromosome, or monosomy, where the cell is missing a chromosome. Say, for example, a germline cell with 47 chromosomes has an extra copy of chromosome 21 and undergoes meiosis to create gametes with that same error. If such a gamete is fertilized by a normal haploid gamete, the resulting zygote will have trisomy 21, Down syndrome. This genetic imbalance can often lead to miscarriage or affect the individual's health, thus negatively impacting the organism's survival. In some contexts, incorrect segregation of chromosomes during mitosis can be slightly positive. As stated before, aneuploidy can cause overexpression of certain competitive traits, which can facilitate adaptation. Because plants can often survive with chromosomal imbalances due to their flexible developmental processes, aneuploidy in this context can positively impact their survival. However, the overexpression of genes due to extra chromosomes can disrupt normal cellular functions, limiting the positive impact of aneuploidy on a plant's survival. Overall, incorrect chromosome segregation during mitosis has a highly negative impact on an organism's survival. Imbalanced genetic material can result in excessive cell death, which can deplete vital cell populations, lead to cancer, and compromise germline cells, which have a negative impact on health and development. While it may lead to the overexpression of certain genes in plants, which can facilitate adaptation, benefiting their survival, incorrect chromosome segregation during mitosis has an overall negative impact on an organism's survival.